This is the second lecture on neurotransmitters and synapses. Hopefully you have reviewed the first one. There may be three just because it's kind of exhausting doing this to an empty room, and I may break it up into three, but it'll still have the same amount of material. So since generally you listen to the first part of the video, I want to tell you a couple things. First off, you need to be checking Canvas. This is the way that they've asked me to communicate with you. Each week at a minimum, you will have lecture video to watch. You will have a quiz that you should be working on, and you will need to participate in a discussion. I ask specific questions in the discussion. It's also the place that I can answer your questions and you guys can answer each other's questions. If we stay better connected, you are more likely to make it through this class. And that's my goal for you, is to make it through this class. So with that in mind, one other thing, and that is that keep using your study groups however you do, but I want to point out to you that there is a Facebook online study group for those of you who are members of Facebook or want to create a fake account just for this Corona time. And it is right here. The SLCC, the SLIC Physiology Online Study Group. So far it's only got 77 members, I think. But the lab aides that aid in anatomy, I've asked some of them to switch over to helping in phys. And for instance, right here, they are offering to do Facebook Live study groups there. So that's a fantastic resource for you guys. There's also just a bunch of lab aides in here that can answer questions, and I'm in here as well. So if they answer something incorrectly, I'll correct it. It's no big deal. There's one of those for anatomy as well, and most of you guys, there's like 600 members in the anatomy one because it's been going on for 2 years. So I just wanted you to know that that existed for you. Okay, so where were we last time? We were just talking about agonist and antagonist drugs and chemicals, and somebody memed the hell out of this. I totally think this is cute. Normal neurotransmitter, receptor, antagonist drugs. And today we get to do one of my favorite things. I am desperately sad that I can't do it with you in the room because I love these conversations that we have. And there's nobody here but me and my dog. Actually, I won't even let the dog in the room because he tends to snuffle and snort. He's really cute, though. The last thing we did was look at ways of theoretically modifying synaptic transmission by drugs and disease. By the way, I only have this up here, not so you can read it, but just so that we can use it to follow along. I realize it makes these images smaller, but most of these images, not the silly ones, but most of these images you will be able to grab from your textbook if you actually need them. So we are going to do neurotransmitters, and as we do neurotransmitters today, we will talk about drugs and diseases, both drugs used for treatment and drugs of abuse, and drugs that are actually kind of both sometimes. So we will do that as we go along. The video that we watched last time is kind of a good place to start. Oops, I hate it when it does this. So, sorry, when I plug in a monitor, it changes the size of those things. I need to remember that in the future. Just be patient with me. Okay. Give me a second. Should restart this, but I'm not going to. Let me restart those little buggers. Okay, so this is what we looked at last time, which is the chemical synapse. And so let's just look at it one more time, and then we'll use this as sort of a jumping off point. So remember,
arriving at the presynaptic terminal.
Remember that basically this is synaptic transmission, and you get an action potential. You get the opening of the voltage-gated calcium channels. Calcium moves into the presynaptic cell, which causes exocytosis of the neurotransmitter. the neurotransmitter diffuses across the synaptic cleft, which is here, and binds to the receptor. In this instance, but not all instances, it is a ligand-gated sodium channel, which would, in this instance, depolarize and cause an EPSP. So that's just to give you a visual to actually get started on what we're doing. So, in addition, we will be referring back to this figure quite often today because this is just sort of a generalized synapse. Okay, so what I want to do is I want to start going through the important / well understood neurotransmitters. You won't learn them all. You'll learn the ones that are in your notes, the ones that people try to modify themselves by chemical intervention or behavioral intervention or whatever the heck they want to do, and the ones that of course ended up being clinically important because of something about their role in the peripheral or central nervous system. So first we'll start right here, which is that neurotransmitters of course are chemical messengers released from what kind of cell goes there. Hopefully you guys know that it is a neuron, and they have to be released close to the receptor. If they're released by a neuron, they go into the bloodstream, then they're actually considered a hormone or a neurohormone, and that's not what we're talking about here. We're talking about neurotransmitters that diffuse microscopic distances and therefore have relatively quick effects, especially compared to hormones. Okay, so what are the characteristics of a neurotransmitter? They are, of course, the language of the nervous system. They are chemicals, ligands, used for communication either within the CNS. So some neurotransmitters only go, for instance, neuron to neuron within the CNS, or between the nervous system and an effector in the PNS. And that would be like at a neuromuscular junction. So acetylcholine in that instance that we learned, the neuromuscular junction, it is a PNS neurotransmitter in that context. Okay, so what do they generally do? Well, generally, by one mechanism or another, they start an EPSP or an IPSP on the postsynaptic cell. Quite often by directly opening or closing an ion channel like we saw in that video with this guy right here. Oh, now it's not going to come back up. We saw in the video, oh, here it is, right here. Sorry. So this one, for instance, was opening a ligand-gated sodium channel, and so it caused an EPSP. But there are other ways of doing that. They can also cause an IPSP if, for instance, instead of binding to and opening a ligand-gated sodium channel, it bound to and opened a ligand-gated potassium channel, it would start an IPSP. IPSP, again, inhibitory postsynaptic, because this is the postsynaptic cell, potential, because it's a change in membrane potential. EPSP, excitatory postsynaptic potential, for the same reasons. Okay. There have been about 60 neurotransmitters that have been identified, probably more than that by now, maybe a few more. More are still being found. I do not have you learn 60 at all because the newer ones we really don't understand what they do very well, so it's kind of pointless to have you guys memorize them. Most of the neurotransmitters are relatively small. What do I mean small? I mean low molecular weight, like in this instance, this is going to be one of the first ones that we look at, which is acetylcholine. If you were not feeling lazy, you could definitely count all of the atoms in this molecule of acetylcholine. By the way, I'm going to show you a bunch of molecular structures today, and I just do that so that you can see similarities between neurotransmitters and sometimes similarities between the drugs that interact with neurotransmitter receptors. I promise you I will never ask you to either recognize it from its chemical structure, or I sure as hell won't ask you to draw it. That's for another class. That's for biochemistry, maybe. So I just show you so that you can have a visual understanding of, for instance, that's what acetylcholine looks like. And this is, hold on, oops. This is how you break down acetylcholine. Hopefully that makes sense. Figure out how to make that bigger. Oh, I can do it this way. But I don't have you memorize the molecular structures. I don't even have them memorized, honestly. Most of them are relatively small, as I mentioned, low molecular weight. And the importance of being low molecular weight is that a relatively fast rate of diffusion across the synaptic cleft with a lower molecular weight. So they're not just huge proteins, for instance. Most of them aren't even proteins. And because of that, they tend to be fast acting. How quickly they act also depends on the type of receptor that they interact with, so we'll get to that a little bit later, maybe today. Often there's more than one receptor for a single neurotransmitter, not necessarily at the same synapse, but for instance, acetylcholine could be excitatory in one place in the brain, for instance. If it bound to and opened a sodium channel, it would cause an EPSP at that synapse. But in another part, acetylcholine could be inhibitory because at another area, if it bound to and opened a potassium channel, then it would cause an IPSP. So try not to rote memorize what they do without looking at it. So I'll try to work you through the logic of it as we go through. So basically, since what the neurotransmitter does depends on the receptor, then the response to a neurotransmitter is really tissue specific because different tissues have different receptors. Okay, so hopefully that'll give you an understanding. Sorry, my nose is itching like crazy. Okay, so now we are going to get into the chemical categories of neurotransmitters. Okay, so when we organize neurotransmitters, we have to look at their chemistry. Not because we need to balance equations or anything, but let me just give you a preview of things to come. Like, why the heck would you care about their chemistry? Well, because in this instance, with this one, do you see that these three right here basically, dopamine, norepinephrine, and epinephrine, do you see how similar they are to one another in structure? So you start from here. This is the precursor to dopamine called DOPA. And add hydroxyl group here, and then add a methyl group right there. Well, why the hell do you care? You care because that means that a lot of times drugs that interact with a dopamine receptor or interact with dopamine reuptake will also work for the other three. So they need to be categorized together so that you can kind of understand what they have to do with their neighbors. In addition, lots of drugs will treat one category of them and not another. All right, so I want to start with the first ones. The first category are called the choline derivatives, and these are derived from choline, which is a water-soluble B vitamin. So these are actually strangely vitamin-derived. And there's more than one, but the only one that you need to learn, you actually already learned a little bit because we used it in the nervous system notes, and that is acetylcholine. And so let's go back to our friend acetylcholine right here. So this is acetylcholine. Again, you don't have to be able to recognize it by look. It is derived from that B vitamin. This is the one that we know a ton about because it was the first neurotransmitter that was identified, and it's really, really common in the peripheral nervous system, and hopefully it makes sense to you that it's easier for us to get a handle on what's going on in the peripheral nervous system because, for instance, you could, without, for instance, causing any kind of scary damage to a person, make the person release acetylcholine and look at its behavior at a neuromuscular junction. Whereas if it was a central nervous system neurotransmitter and you wanted to mess around with somebody's dopamine levels, A, it's really difficult to do that, and then B, the response is potentially unpredictable or unpleasant, and I certainly wouldn't volunteer for it. It is a common PNS neurotransmitter, but it also has some CNS functions. So each time I'm going to go through functions, receptor, removal, and then sometimes there'll be clinical connections. So I want to point this out to you as well. I made a table for you. I was going to leave it blank. That's what I usually do, but under under these circumstances, I filled it in for you. And so if you click right here and go to the neurotransmitter, this is a student version. This has the same kind of stuff that we are looking at. You're only responsible for stuff in this table that is also in your notes. So it's kind of like a way to organize the neurotransmitters like you did with the hormones, but there's not very many of them. See, it's not that many. It's not like 32 different ones, and there's no feedback loops or anything to memorize. And it's all in your notes too, but I'm kind of a table person, so I like it in a table, and you can reorganize it as you see fit. All right, so acetylcholine functions. So what does acetylcholine do? Well, it has a few different functions. It is usually an excitatory neurotransmitter, simply meaning that it usually binds to excitatory receptors. And the terminology that goes along with acetylcholine, the easy way to say that something responds to, uses acetylcholine, is this word right here, which is cholinergic. So do you see this word right here? So this is the easy way for someone to say this synapse right here, is a cholinergic synapse, meaning that it responds to, has receptors for acetylcholine. Heck, what happened? So it releases acetylcholine, it has receptors for acetylcholine, it reuptakes or destroys acetylcholine. The big ticket word for all of that is cholinergic. Okay, so this whole synapse would be called cholinergic. The synapse is cholinergic, everything's cholinergic there. So let's talk about some CNS functions and PNS functions of acetylcholine. So the PNS function, peripheral nervous system function of acetylcholine, you already know one of them. Skeletal muscle, all of those neuromuscular junctions for skeletal muscle in the somatic nervous system, those are all cholinergic synapses or cholinergic neuromuscular junctions, all of them. And then right after this set of notes, we will get to the autonomic nervous system, which is just a short little set of notes. Most of the autonomic nervous system is cholinergic as well. So they all use acetylcholine. So those are PNS functions, right, between the neuron and the muscle, or the neuron and the gland. But there are some CNS functions of the acetylcholine, meaning not only is acetylcholine released between a neuron communicating to my skeletal muscle to make me do this, but it's also released between neurons and my brain in specific regions of the brain. So one of the CNS functions we have found for acetylcholine is that it's used in short-term memory and learning, and we'll get back to nicotine in just a second about that one. So CNS functions, short-term memory and learning, and that's acetylcholine. So what do you call the receptors for acetylcholine? So this guy right here, notice this is a cholinergic receptor. And this cholinergic receptor right here means that it responds to acetylcholine. That's just a general term. There are going to be two different subcategories of cholinergic receptors that you'll run into this semester and in your future. And the subcategories of cholinergic receptors are the nicotinic cholinergic receptors, called that because we found them because they respond to nicotine, but they are evolved to be cholinergic receptors. So we called them nicotinic cholinergic receptors. And then there are also muscarinic cholinergic receptors, and the muscarinic cholinergic receptors respond to acetylcholine. And they also respond to a mushroom poison that was killing people, and they had to figure out why. And that mushroom poison is called muscarine, hence muscarinic cholinergic receptors. But they're both cholinergic receptors. It's just two different categories. So what is the primary way that acetylcholine is usually gotten out of the synapses, because we know it's really important to get them out? And the primary way that that occurs is usually by an enzyme that you already met, which is acetylcholine esterase. a few places in the body they actually do reuptake of the choline portion gets reuptaken. So acetylcholine esterase is really, really important in cholinergic synapses. And what it does is this. It breaks acetylcholine down to acetate, acetate and choline. And then acetate is the same thing as acetic acid. And then it reuptakes the choline, which is the B vitamin derivative, so that you can, for instance, use it to make more acetylcholine. So it doesn't reuptake the whole thing, and just reuptakes one of the breakdown products, which is choline. So it reuptakes part of it. Okay, nicotinic, muscarinic. And as far as clinical connections that go along with acetylcholine, so I don't know if you've ever heard of, I don't know if I mentioned this before, there's a poison called curare that is derived from South or Central American plant, and it's used on blow darts and arrows and things like that. And it's an acetylcholine antagonist, I think I have mentioned it, at the neuromuscular junction. So remember, antagonist means that it blocks the receptor and does not allow the cell to have a normal response. So an antagonist would basically sit right here and keep acetylcholine from binding right there. And then nicotine. So again, back to why people like nicotine. People like nicotine because it is an acetylcholine agonist, meaning mimic, in the central nervous system. And since acetylcholine in the central nervous system is for short-term memory and learning, using nicotine initially makes you feel kind of with it, kind of like you're firing on all cylinders mentally. That effect doesn't last forever, but that's part of the reason that people like it initially. And then the other thing that is related to acetylcholine, since it's acetylcholine in the central nervous system is related to long-term memory, short-term memory and learning. Not surprisingly, Alzheimer's has an acetylcholine component. There are portions of your brain that lose the ability to produce adequate amounts of acetylcholine for reasons we're still trying to understand. And those regions just start to degenerate, and you don't have as much synaptic plasticity. The cells aren't communicating with one another to the point that post-mortem is the only way that Alzheimer's can truly be diagnosed. But you can see the tremendous loss of primarily gray matter that occurs with advanced Alzheimer's. So that is acetylcholine. So that's your first one, and it's the only one that fits into the category of choline derivatives that you are going to learn. Okay. The next ones are called biogenic amines. That's the next chemical category. And so what these are is they are derived from amino acids, but they are changed so that, for instance, they can't function as an amino acid any longer. So again, I'm just showing you molecular structures to help bring things home, not because you're going to be responsible for them. So here's an amino acid called tyrosine. And tyrosine looks like this, okay? But then when I get my first true amino acid out of it, do you see it doesn't look very different than an amino acid? And then these two don't either. So what a biogenic amine is they're synthesized from amino acids, but they're changed enough that you couldn't use them as part of a peptide. So about the same size as an amino acid, derived from amino acids, but biogenic, you changed them. Okay, so there are three. There are four, but there are three in the first category. So the first category of biogenic amines are here, which is the catecholamines, and these are all catecholamines. So there are basically going to be three catecholamines, all derived from the amino acid tyrosine, and they are going to be dopamine, norepinephrine, and epinephrine. They are going to be very, very similar to one another chemically, just small changes, change. And so, not surprisingly, they will have often similar receptors, often similar uptake or removal mechanisms, and that's going to be important to you guys, and just for having a brain, basically. Okay, so let's meet them. So the first one is dopamine, which might, without your knowledge, be your favorite neurotransmitter, because dopamine is involved in this happy little triad of neurotransmitters that makes you both happy and sane simultaneously, which is admittedly a difficult thing to accomplish right now. So dopamine does multiple things. One of the things that it does is that it is used in the brain, central nervous system, for motivation and reinforcement of behavior, so reward and motivation. It is your do that again pathway. It tends to feel really good evolutionarily when you did something like eat a really sugary food or fatty food. Your brain said that was amazing, do that again and frequently. And it made you want to do that because dopamine feels really good, so you tend to seek that out again. And so it tends to be related to food, to sex. Some people release dopamine with exercise. I am not one of those people. And then, not surprisingly, drugs that tap into the dopamine pathway tend to reinforce the drug. It says, that was awesome, but instead of just saying that was awesome, it says, that was awesome, do that again and frequently. So that's really important to understand with the whole reinforcement and perseveration pathway. So in a different portion of the brain, so the portion of the brain involved in the reward and reinforcement pathway is like within the limbic system primarily, but there's a different portion of the brain that in which dopamine is also involved in voluntary movement. So your cerebral cortex controls voluntary movement, but it has to send it out through a region called the substantia nigra. And the substantia nigra is deep within the brain. It's one of the cerebral nuclei that's like buried deep within the brain. And the substantia nigra has to be functioning for that command for voluntary movement to make it out to your muscles. And when that starts to go bad on you, you can actually start to demonstrate Parkinson's disease. So Parkinson's disease is basically a loss of the ability to produce adequate amounts of dopamine in that particular portion of the brain. But then what else is related to dopamine? Obviously addiction is related to dopamine, because this reward and reinforcement pathway and the pleasant nature of the release of dopamine tends to reward you for doing, for instance, drugs and chemicals that interact with the dopamine pathway. So just because you're probably curious, I've got a huge neurotransmitter spreadsheet, but I'm not going to overwhelm you with it. So the location function, so you can kind of see these: reward, pleasure, reinforcement. Hold on just a second. So not surprisingly, drugs that tend to be really high on addiction predisposition. Sorry, I'm trying to make this big enough so that you can see it, just because you'll be curious. Those drugs, not all of them, but most of them, cocaine, amphetamines, including methamphetamine, those are really, really big on reinforcing themselves in the dopamine pathway. And then, interestingly, and we'll talk about this later, secondarily, not primarily, but secondarily, the opioids, which we'll talk about a little bit later, don't work with dopamine directly, but generally speaking, when you take opioids, they activate the dopamine reinforcement pathway. So even though that wasn't the reason you were taking them, that's the reason people often get addicted to them. All right, so the other thing that they have figured out that is related to dopamine is schizophrenia. And schizophrenia has a very strong genetic component, and usually manifests in early adulthood. And what happens with schizophrenia, it seems as if a portion of your frontal cortex is actually over-releasing dopamine. And that manifests as these schizophrenic behaviors and schizophrenic thought patterns. Really difficult to treat that one. Okay, so that is dopamine. That's what you need to know about dopamine. And then norepinephrine and epinephrine are the two that are very closely related to one another, also very closely related to dopamine. I usually lump these two together, even though some receptors only respond to one of them. But as far as norepinephrine and epinephrine, norepinephrine is mostly a CNS neurotransmitter, and epinephrine is CNS and PNS neurotransmitter, but for our purposes, they do something very similar. And of course, when we learned hormones, we also learned that epinephrine was also a hormone called adrenaline. Okay, so what do norepinephrine and epinephrine do? CNS and PNS functions. CNS function, let's go back to our happy Venn diagram here. The CNS function of norepinephrine is really kind of oversimplified as alertness, concentration, and energy. Think of it as an up neurotransmitter, not spazzed out, but I am aware, I am paying attention, I can do this, I've got some adequate level of energy to do it. And oh, I forgot to say that. Oh no, sorry. I forgot to say one thing related to dopamine. So the receptors for dopamine are called dopaminergic receptors, and there's actually two categories of them, D1 and D2. I don't usually test you on D1 and D2, but if I remember correctly, it's the D2 receptors that tend to be highly related to addiction. And so they work really hard at trying to find something that will, for instance, activate the dopamine response without activating the D2 receptors. But back to my original path, which was talking about norepinephrine. So our friend norepinephrine. Norepinephrine is an epinephrine. Any cell, as far as the terminology goes with these, any cell that, or any receptor that releases and responds to norepinephrine is called, or epinephrine is called adrenergic. So what I want you to do for just a second is to pretend that this synapse right here is specific to, let's say, norepinephrine. You would call the whole thing adrenergic, and I realize it would make more sense if you called it norepinephrinergic or epinephrine. But when they found these chemicals, they actually tracked them back through the bloodstream to the adrenal glands, because of course you release these from the adrenal glands as well. And it's way easier to find a hormone than it is a neurotransmitter, because hormones circulate in the bloodstream. So they assumed that these always had to do with the adrenal gland, and they called everything related to epi, norepi, adrenaline, all of it was adrenergic. And then when they found that they were also present in the brain, they didn't change the name of them. So again, epi and norepi, anything related to them, assuming this was actually norepinephrine or epinephrine synapse, we would call this an adrenergic synapse. There are two different kinds of adrenergic receptors, alpha and beta. Those end up being important later this semester when we talk about, for instance, cardiac muscle, which has beta adrenergic receptors. What do these guys do? What these guys do is, back to our picture here for a second. Okay. So to oversimplify the function of these guys, their CNS function is primarily like arousal, not sexual arousal, but just like awareness, arousal, attention. They have something to do with mood as well. But they also are released in the peripheral nervous system. Specifically, they are related to sympathetic nervous system responses. The sympathetic nervous system will use mostly norepinephrine to cause the fight or flight responses, and we'll get to that in the next set of notes. So to reinforce one more time, these two right here, norepi and epi, those go together. I just lump them together for my purposes. They are CNS and PNS. Anything related to those, if for instance we go back to this guy right here and we said this was a norepinephrine synapse, the quick way to say that it releases, responds to all of that stuff, norepinephrine or epinephrine, would be to call the whole blasted thing adrenergic. And functionality of these is primarily attention, arousal, has a little impact on mood as well. And then PNS function is fight or flight. Now, one more time, back to this. These three, dopamine, norepinephrine, and epinephrine, all together, those little guys are these guys right here. These guys are called the catecholamines. They are biogenic amines that are specifically derived from the amino acid tyrosine right here. They all go together. Because they all go together, they will not surprisingly have a similar removal method. Catecholamines are removed at some synapses. At some synapses, they are removed at some synapses, they are removed by reuptake, and at some synapses, they are removed by an enzyme that breaks them down. So let's look at that. So reuptake is obviously right here. So this is a reuptake. Damn it. Sorry, I'm trying to get this tool down, and apparently I have some work to do. Removed by a reuptake mechanism. And that means that if you, for instance, feel like you would feel a little better if you had more dopamine in your brain without, for instance, taking cocaine. One of the ways that you might accomplish that is to block the reuptake of dopamine. So there are drugs that we'll talk about that will actually do that. And then another way at other synapses is that in these catecholamine synapses, there's an enzyme that breaks down dopamine, norepinephrine, and epinephrine at the synapses, and that enzyme is called monoamine oxidase. So in this instance, this would, for instance, be monoamine oxidase. So this enzyme right here that would break down dopamine, norepinephrine, and epinephrine is monoamine oxidase. At different synapses, it's actually reuptaken. So the reason that's important is because that brings us to the drugs that we can use to treat those two chemicals that are part of the happy triad. So let's go back to our happy triad here and say, okay, I went in to my physician and I said, I'm feeling kind of down. And this was before it was a pandemic, because it's a totally reasonable time to feel down right now. And what could you do under those circumstances? Well, what you could do is you could decide if you wanted to, for instance, treat norepinephrine, maybe you'd be a little more up if you did. Treat dopamine, maybe you'd be a little happier, a little more rewarded, a little more happy with your life. Or quite often they choose this one, but let's talk about these two for just a second. So one of the ways that people might choose, and their healthcare provider might choose to treat, for instance, depression, is with a drug that manipulates your norepinephrine and or dopamine levels. Wellbutrin, which I can never pronounce the actual name of it, is bupropion. What it is, and I want you guys to see if you can kind of grasp what I mean by this. Wellbutrin is a dopamine and norepinephrine reuptake inhibitor. So what it would do is it would allow the dopamine and norepinephrine to stay in the synapse longer. And when it stayed in the synapse longer, what it would do is cause greater activation of the postsynaptic cell, which is basically fooling your body into thinking that you have more dopamine, hence I'm happier, and more epinephrine or norepinephrine, hence I feel more up. And that is certainly a form that might feel like a relief from depression. What that particular category of drugs is not good for, hopefully this will make sense, and it's contraindicated for anxiety. So anxiety and depression sometimes have similar chemical reasons, but some drugs are effective for both, and some drugs are not quite as effective for both. So these drugs that increase the amount of norepinephrine in the synapse sometimes have all of the benefits, but then they cause sleeplessness and maybe a little bit more anxiety. And if you are predisposed to panic attack, they're definitely not recommended for that. And that is, for instance, Wellbutrin. It's called an SNRI, not an SSRI. I don't know why it's not an SNDRI, but it actually works on norepinephrine and dopamine. The other category of drugs that does something similar is not used very often, so I'll just tell you about it in theory, and that is, back to our picture for a second, that is a drug that works by inhibiting the enzyme monoamine oxidase that breaks down dopamine, norepinephrine, and epinephrine in CNS synapses. And so it's basically a blocker for not the receptor, but for the enzyme. And so it kind of has the same impact in that dopamine and norepinephrine stay in the synapse longer. So this, they're used relatively rarely as antidepressants. The two that I know of are Nardil and Marplan. And from what I understand, and if you guys know something different, will you put it in the discussion? Because sometimes some of you guys work in psych and know these things that I don't. And I like, part of the reason I miss you is because you guys teach me stuff too. Those are not used very often in this country. And they're also, as far as I understand, not used very often in outpatient situations. And the reason that I understand that that's the case is not because they're ineffective. They're actually pretty darn effective for treatment-resistant depression, but they have a suite of side effects that are really difficult to manage because the side effects are really common when you interact with some things that people ingest regularly. Like, for instance, they have this really negative interaction with cheese. Oh my goodness, I think I'd be more depressed if I couldn't eat cheese. And wine, heavens, how are we supposed to live like this? And some over-the-counter medications. So quite often when they're used in inpatient situations, and I still don't think they're used very often in this country. But again, if you know something, teach me in the discussion portion. Okay, so that is all of the biogenic amines. I'm sorry, all of the catecholamine category of biogenic amines. And then the next, these are also biogenic amines. And the one I want to talk about is serotonin. And I know you guys have heard of serotonin. And I'm wearing serotonin on my neck, by the way. Seems like a good time. You can't see it. Seems like a good time to have a little bit more serotonin. Doesn't work this way. So other biogenic amines. Serotonin is synthesized from a different amino acid. So it's not a catecholamine because it's not synthesized from tyrosine, but it is still a biogenic amine because it's really similar to the amino acid that it comes from. So it's actually synthesized from the amino acid tryptophan. And what it really is tryptophan with a hydroxyl group on the 5 carbon. So it's called indolamine or sometimes 5-HT because 5-hydroxytryptophan. Tryptophan with a hydroxyl group on the 5 carbon. hopefully that makes sense to you, to those of you who like chemistry. For the rest of you, the only reason I tell you that is because sometimes instead of calling the receptors serotoninergic, which is great when they do that because you know it responds to serotonin, they call them 5-HT-ergic because 5-hydroxytryptophan is what serotonin is. Okay? All right, so what does serotonin do for you? It's a really broadly used CNS neurotransmitter. It is used all over the brain in some really important ways. It's used in the brainstem, and the brainstem has to do with sensation and some motor control. It doesn't work on the parts of the brain that control your heart rate or anything, but brainstem. It's usually an inhibitory neurotransmitter. Look at all the functions of serotonin. It regulates sleep, not alone, but with friends. Not sleeping with friends. I'm talking about it. There are other neurotransmitters that help as well. It also regulates emotion, but because of its brainstem effect, it also regulates sensory and motor function. It regulates hunger, mood, sex drive, anxiety, obsession, compulsion, and memory. So let's look at where it fits into our happy triad. So if you've got it alone in the wrong amounts, then it's basically those guys. If you've got it in perfect Goldilocks amounts, which would be lovely, then what it does is works with the other two so that you get this perfect middle of the Venn diagram, which means I am up enough, I am happy enough, and I can manage. If your serotonin is working rather well and you have the proper amounts of it in the proper place, you, for instance, can roll with the punches. So when you have a pandemic, you might not completely break down on a regular basis. Or under other circumstances, if your serotonin is very low, it can manifest as anxiety. It can manifest as depression. Apparently, anxiety is slightly more common with low serotonin in women, and depression is slightly more common with low serotonin in men. This happy little portion right here, I've probably been there once or twice in my life for like a couple minutes at a time. And it's definitely worth the battle to get there, but it's pretty much just constant. Okay, so what is the most common way that serotonin is removed from synapses? Back to our picture. So how do you get serotonin out of synapses? The most common way that it's removed from synapses is by a reuptake mechanism. So it's dumped into the synapse. Let's say this is a serotoninergic synapse, and then I get rid of it by reuptaking it, which brings us, of course, to our category of drugs that we all know about. Very high rate of prescription of these in Utah, even compared to the rest of the country. And these drugs are used for anxiety and depression, and they're kind of a first line of medical intervention for anxiety and depression. And they are called the SSRIs. And that is right here. SSRIs are selective serotonin reuptake inhibitors, SSRIs. And they are used both as antidepressants and as anti-anxiety medication, whereas the SNRIs, like Wellbutrin, are more likely to be used as antidepressants and not anti-anxiety medication. So I want to show you a little bit about sort of the history of their use. And it doesn't go current, but I just love the image. Hold on just a second, let me find it. All right, so that's what serotonin looks like, by the way. It's the one that is on my necklace. Okay, so let's look at the SSRI antidepressants and figure out. So this is like the expanding repertoire, but this only goes to 2005. I just loved the visual, and they hadn't remade it. So I realized this is 15 years old, but look at how cool it is. So what we would like, if we have an SSRI, is for it to be selective to the synapses. I told you it's used all over the brain, but I would prefer that it doesn't affect hunger. It doesn't necessarily affect sleep. I want it to affect mood. I don't necessarily want it to affect sex drive. So the more selective you are, the less side effects you get from using this medication. So the selective serotonin reuptake inhibitors. The first one, which is Prozac, all the way back here in the late 80s. So it's called Prozac. And then these are the things that it's approved for, all of these in pink, and when it was approved for that. Depressive disorder, and then obsessive compulsive disorder, and then bulimia, PMS, panic disorder, all of those kinds of things. So it's been used for those. But it wasn't very selective. So although it was really effective, it didn't have... no. Although it was very effective, it had lots and lots of side effects. And the side effects... Oh, we're having a dog party in here. Hey, go on, guys. Go on. Okay. Although it was very effective, it had a lot of side effects. That was Maddie and Superfly. Maddie is almost 13, and Superfly is 1.5. And they always want to get in when I'm doing this. Okay. So although it was very effective, it had a lot of side effects. So it wasn't selective enough. It's still really effective, but it has a lot of side effects. Some of the side effects, because of the functionality, would be changes in sleep patterns. One of the big ones that people don't like is that it tends to cause a lot of weight gain and tends to crash the sex drive. So the next one that's developed right here is Zoloft, and it's in the green here, and you can see it's approved for these number of things as you go along. That one was slightly more selective, and then as we move on, we start to get things like Lexapro and Citalopram, Escitalopram and Citalopram. These are sort of the more selective of them, but they don't work on quite as many people, but when they work, they have fewer side effects. So fewer undesirable side effects. So we have this whole suite of SSRIs, and there are more than these now. This is just this image that I really like. I wish I could figure out where I got it so that I can find the updated one. But anyways, those are your SSRIs. Note, though, that when, for instance, you go into your healthcare professional and say, hey, I've been sad, depressed, have all these symptoms, and they say, oh, okay, well, maybe we'll try to treat you with a medication. They don't ever actually assess your serotonin level. They don't assess your norepinephrine level or your dopamine level. They just give it a shot. And for reasons we don't completely understand yet, the SSRIs actually take a while to start working, and then if you go off them very quickly, you're going to have to deal with a massive amount of downregulation of the receptors. So if you thought you were depressed before you went on an SSRI, if you go on an SSRI and then go cold turkey off of an SSRI, you are going to have both low serotonin and insensitive or too few receptors, and this is definitely not recommended. You have to step down off of these things, especially the SSRIs. For some reason, the SNRIs work a little more quickly, and you can get off of them a little more quickly. But when you feel fine and you decide you want to go off of an SSRI, cold turkey is not the way to do it because of the whole downregulation risk. So these are our SSRIs. The other things that are kind of interesting about our friend serotonin is that serotonin and drugs that interact with serotonin receptors are also relevant in some other things. For instance, almost every hallucinogen that we've ever found, LSD, psilocybin, those all interact at serotonin receptors. Some of them are serotonin agonists, some of them are not. And so you get all of these changes in like emotion. You can get changes in sensory function, which of course causes hallucinations, changes in motor functions like tingling. They can impact your hunger, your mood, your sex drive, your anxiety. These are not at all selective. They basically go in serotonin receptors all over the brain. And LSD, for instance, is really, really dramatically effective in microgram quantities. And psilocybin is naturally derived ones from fungi and some other things in nature. MDMA, which is ecstasy, also called Molly, this one is related to serotonin because what it does is, going back to our serotonin story, back here, let's go and pretend that this synapse is serotonin for a second. What ecstasy does is causes a massive dump of serotonin all over your brain, and so you tend to feel really good because serotonin will make you feel really good and very open. But it also inhibits reuptake of serotonin, and since it's inhibiting reuptake of serotonin, it gets dumped and then it stays in your synapses for a long time and continues to stimulate the serotonin response on the postsynaptic cell. So you feel amazing. For a while, you might have a little hallucination, you might have some sensory function, but you don't reuptake your serotonin, which means you kind of dumped it. And a couple of days after that, you are going to feel really, really depleted on serotonin. So because it was used as a club drug, of course, in the 80s, 90s, even in the thousands, and people would use it on Saturday, and they would use it like a lot. They would call a few days later Suicide Tuesday, because you would feel so sad because you were so depleted on serotonin that you didn't feel like you could function. Now, one of the things that might be interesting to you guys, I can't remember if I linked it at the bottom of these notes, I'll look in a minute, is that MDMA is now, it's been Schedule 1, meaning that there's no approved use and it's illegal in this country for a long time, since the 80s. But they have started some clinical trials with MDMA with therapy at the same time, specifically for post-traumatic stress disorder, which I think they call PTSS now, post-traumatic stress syndrome, or post-traumatic stress response. I can't remember. But anyway, they've been using it, and because it tends to bring down your barriers if you use that in combination with talk therapy. And of course, there's a physician or a nurse present, and they would use MDMA-assisted therapy once per month, and I think I'm remembering this right, but I'll try to link the article, once per month with a therapist and a medical professional there. And then they actually go to therapy every week for a month, and they do this for 4 months. Because of the lowering of barriers and the combination with being able to talk through it, it has been incredibly effective for helping people get through resistant PTSD, which tends to be very resistant to therapy. And first they used it with violent crime survivors, and now they're using it with soldiers. I think we're in the third set of clinical trials on it. So it'll be really interesting to see what happens with that. I mean, you probably won't be able to get it in Utah anyway, but probably at some other places in the United States. And I know you all have the textbook now because I got you free access to it. So there is a box in your textbook called Treating Depression. It's totally worth reading. It talks to you about the different ways of treating depression. Okay, and then the very last one right here, which is a biogenic amine, is called histamine. And the only reason I want you to know about that one is because histamine you learned before as a paracrine agent, because antihistamines, but it's also a CNS neurotransmitter that we might end up being important because it's used in the hypothalamus, which is arguably pretty important for physiology. And that's really all you have to know about it, because we don't really know very much more about it yet. Okay. Next category of neurotransmitters are the amino acids. By the way, if at any time you want to stop when you're watching these videos, just take a break. I would like to take a break right now, but I want to get it done. So the next category of neurotransmitters are straight amino acids. And unlike biogenic amines, in which I take an amino acid and I change it so that I couldn't use it as part of a peptide anymore, with the amino acids, it's just a straight amino acid that's actually used just as a neurotransmitter instead of as part of a protein. And so the 4 that you guys are going to learn are these 4. So there are 2 that are generally excitatory, and there are 2 that are generally inhibitory. So glutamate and aspartate are amino acid neurotransmitters that are generally excitatory. GABA, which is gamma aminobutyric acid, but GABA is fine. I won't make you spell that one out. And glycine are amino acid neurotransmitters that are generally inhibitory. So let's see those. Okay. So amino acids, by the way, are the most abundant of the CNS neurotransmitters. I'm only having you learn 4 because we know the function of these relatively well. So the 2 excitatory ones, the ones on the top, glutamate and aspartate, those 2, glutamate is primarily used in the cortex, the cerebral cortex, and in a portion of the brain called the hippocampus. And what this one is for is neuroplasticity. So neuroplasticity means your ability to, for instance, make some new synaptic connections. So especially in the hippocampus, which is for memory, and especially in the cerebral cortex. So it's probably a pretty important neurotransmitter. So this one right here is implicated in ADHD. And it's also implicated with another friend that we already learned about, dopamine. It's also implicated in Parkinson's disease. There's a drug that I don't know if anybody does anymore, what do you call them? Like a street drug. I don't even know if it was developed for a medical purpose. But PCP, PCP is that drug that you used to hear about. You know, I remember the 80s. Some of you guys might not, but when you heard about some guy acting crazy and it took like 6 cops to bring down a 150-pound dude, and he felt like he was 10 foot tall and bulletproof, that is PCP, which interacts at glutamate receptors. And then have you heard of ketamine? Ketamine is also, it's a glutamate antagonist. So it's an antagonist, an excitatory neurotransmitter. It's used as a tranquilizer for animals, also sometimes for humans. But it's also used as like a drug that people just take for kicks and giggles, and that's ketamine. Ketamine is also in clinical trials as a powerful candidate to relieve treatment-resistant depression. So there's some interesting stuff that's going on with that now. Aspartate, which is this guy right here. Aspartate kind of does the same things as glutamate, only this one is mostly brain and this one is mostly spinal cord and brain stem. So this one's like higher brain and this one's like lower brain and brain stem. And it's kind of like glutamate. The receptors for these 2 are called Nmda receptors. And I won't make you spell out NMDA, but they medically they tend to be important because, like I said, one of them is used as an anesthetic. So NMDA receptors. All right, so these 2 right here, GABA and glycine. GABA is my favorite. GABA is gamma aminobutyric acid, but I promise you I won't make you spell that one out. Just call it GABA. Everybody calls it GABA. Its CNS function is that GABA is nature's chill-out chemical. It's nature's Valium. It makes you feel very calm. The receptors for GABA are called GABAergic receptors. And the reason you may know about this one is because a lot of the anti-anxiety drugs, not antidepressants that are also used for anxiety, but specifically they call them anxiolytic drugs, most of them actually work at GABA receptors. So GABA is your chill that you feel. So some drugs that work at GABA receptors, Valium, and all of the whole category called benzodiazepines, quite often just called benzos, including Klonopin, clonazepam, lorazepam, all of those, the PAMs. Those all enhance the action of GABA, so they make you feel more chilled out. Alcohol, ethanol, also enhances GABA's action. So if you take a sip of wine and then a couple of minutes later you feel your shoulders relax and you feel your blood pressure drop and you feel that calm that comes with small amounts of alcohol, large amounts of alcohol actually tend to jack this all up, by the way. So these also enhance the activity of GABA. Now, because these both, your benzos and your alcohol, both enhance GABA's action, using them together tends to be very, very dangerous because you can relax yourself to death. So you don't want to use benzos and alcohol simultaneously. There's also another box in your text about the role of GABAergic agents, these including benzos. It's actually really interesting to read about that in your text. This is all in chapter 8 of your text, by the way. And then last of the amino acid neurotransmitters is glycine. Glycine does similar things to GABA. GABA is mostly higher brain. Glycine is mostly spinal cord and brainstem. Strychnine, by the way, is a poison that kills you because it actually blocks glycine's action. So it's a glycine antagonist. Okay, we're almost finished with neurotransmitters. Just a couple more to do. Okay, so now just a tiny little bit about the purines. There is a whole category of neurotransmitters called the purines. As in, remember learning in biology, purine, pyrimidine, pairs that make DNA, and then purines and pyrimidines also, and RNA. So what are the purines? It's kind of a bad picture. Hold on, let me. Okay, so I don't need you to memorize this, but this is ATP. ATP is adenosine triphosphate, and so it's got 3 phosphates. This is ADP, this is AMP. If it would just adenosine, it would be that without the phosphate on it. So you know, of course, that mostly these are used either as portions of nucleic acids or as energy molecules, but surprise, surprise, they are also used as neurotransmitters. And the only reason you care is because A, it's a little weird to know that ATP and ADP can be released at synapses to cause postsynaptic responses. So this could be like ATP released instead of acetylcholine or something, or ADP or adenosine. And the only other reason that you might care is because caffeine also works on these. Caffeine is actually an adenosine. So imagine, I couldn't find just adenosine and the others in the same picture, but just caffeine is actually an adenosine antagonist. And let me just say this. Adenosine is a neurotransmitter in the central nervous system that is generally inhibitory. And so an adenosine antagonist, if you block the effect of an inhibitory neurotransmitter, you get excitation, and so you get your buns out of bed in the morning. So that's the reason I thought you guys might think that one was interesting. Okay, and then the last real categories are the neuropeptides, and neuropeptides are big uns. So this is a neuropeptide way bigger than the ones we've been looking at. Still relatively small. Look at all of these amino acids. So what they are is a short, relatively short. I mean, I know it's long compared to what we've been looking at, but it's really short compared to most peptides. It's a short chain of amino acids between 5 and 40 amino acids. They are larger, so they tend to be slower acting because they diffuse across the synaptic cleft a little slower than the other neurotransmitters. And so these are important because this is going to be really important to you guys because you guys are going to go into healthcare, right? So these are your endogenous. Endogenous means that you make it, okay? opioids. These are the opioids that you make in your body. And of course, the opioid drugs that we take for instance, pain management, are going to interact at receptors for your endogenous opioids. Okay, so it's going to, let me teach you the ones that you make first, and then I'll teach you a little bit about the drugs. Okay, so the endogenous opioids include your naturally occurring opioids. One of those that you might have heard of before is called an endorphin. So endorphin lowers your pain, makes you feel 10-foot tall, bulletproof, you feel kind of good, right? So what do? Another one is, by the way, enkephalins, and enkephalins is similar to, but not the same as endorphins. What they do is in the CNS and in the PNS, they act as natural opiates, and they reduce pain perception. Okay, so let me try to show this to you on the video. There are 3 ways. Where's your arm, Melanie? Okay, there are 3 ways I could potentially treat pain. Let's use this hand. Okay, so I could treat sensation of pain, right? Stop pain at the sensory receptor, right? I could treat transmission of pain, keeping the signal from going in to the central nervous system. Or what these do is treat the perception of pain, which happens in the central nervous system primarily. So they reduce pain perception. So they tend to act on your CNS. The receptors, what we call these is we call these opioid receptors because we actually found opium long before we found neurotransmitters. And so we named the receptors after the drug instead of after the actual neurotransmitters. So we found opium. People started smoking opium, taking opium in different ways. And they're like, this is amazing. I want to continue doing this, right? And of course, we'll talk about the addiction nature in just a second. Then fast forward thousands of years before we actually found the neurotransmitter system that they were actually interacting with. So the receptors are called opioid receptors, and they include delta, kappa, and mu. The only one that I think that you would really care about is this little guy right here, because the mu1 receptor seems to be the one that's most directly related to dependence. So if we can come up with drugs that treat pain but don't activate the mu1 receptor, that would be golden to be able to do that. And you know, it's still in the works. We're still trying. Okay, so of course the clinical connection is exactly what you thought it was going to be. Opioid drugs, right? So what can I tell you about opioid drugs? Because these are natural pain relievers, we can have drugs that treat pain that are based on this neurotransmitter system. And so we have morphine and heroin and codeine and OxyContin, et cetera, et cetera, et cetera. And all of these are either derived from opium or synthetics that act like they were derived from opium, and they interact at specific opioid receptors. They tend to be highly habit-forming because what happens when you take these is, in addition to activating the opioid receptor, it also causes activation of the dopamine pathway and tends to reinforce itself. Opium use, the earliest that I could find that it was recorded was 3400 BC. So that's how long people have been using these opioid drugs. So the other thing that tends to be important with these is that not only are they highly habit-forming, but they are also, if you continue to take them, even if you continue to take them for pain relief, they cause a downregulation of receptors, which causes a downregulation of both the pain-inhibiting response and the pleasure response. So the net effect of that is that people tend to seek out more or different opioids to both get a reduction in pain and an increase in pleasure. So when opioids are released, dopamine is also released in this portion of the brain called the nucleus accumbens. That is the habit and reinforcement. So the take-home message is, even though you weren't trying to mess with the habit reinforcement pathway when you take opioids, you can. How likely are you to become addicted to these things? It seems to have a relatively strong genetic component that you might not know until you mess with it. Okay, so let me just show you a little bit about opioid drugs. I thought this was really interesting. So these are the rise in 3 waves in the rise of opioid overdose drugs. Oops, come back here. So heroin, by the way, was created to get people off of morphine, and that was an oopsie because people really like it. So we've got all the way back to 1999, we have the commonly prescribed opioids, and then those get less available, and then heroin is produced relatively cheaply, and we get a rise in heroin use, rise in heroin overdose deaths that occurs right there. So this is the wave in prescription, and then here's heroin. And then somebody creates drugs that are way the heck stronger, and like fentanyl. These are either prescribed or illicitly manufactured or sometimes imported, and they need so much smaller quantities to actually kill you that you can see this massive rise in synthetic opioid deaths. That is all from 1999 to 2017. So here's yet another epidemic in addition to the one that we're currently experiencing. So one last little tidbit about neuropeptides and opioids, and that is that sugar addiction doesn't seem like it would be a real thing, but if you were a type 1 diabetic, it definitely could be. So just as we're all feeling very, very superior, I want you to know that sugar addiction can be circumvented or interrupted by the same set of drugs that they use to treat opioid addiction. So you just may be addicted to something that's socially acceptable. Okay, a couple more that people told me they were interested in. I don't usually ask about either of these, but CCK is a neuropeptide that regulates gallbladder contraction, tends to be important for people with gallbladder problems. And then this one called orexin is a neurotransmitter that's in the hypothalamus. It helps to regulate your sleep-wake cycles primarily by controlling what the pineal gland does. And this is used as a potential treatment for narcolepsy. And then the last one is there's this neurotransmitter that was discovered recently called substance P. And substance P is involved in sensing pain. So we said sensation, transmission, perception. This one is involved in sensing pain and may be an analgesic or a pain reliever that we might be able to use in the future that doesn't tap into the dopamine pathway like opioids do. But still, you guys know how common opioid, both prescriptions and addictions are. And then the last big ones, and then I will stop before we get to receptor functions because I'm tired and you're probably tired listening. So this category aren't even in your textbook. I don't know why. But I think you guys will think they're interesting because, of course, this is culturally relevant and relevant to legislation right now. And these are called the endocannabinoids. And the endocannabinoids are the ones that are related to, for instance, cannabis. So we didn't know for a super long time what they did, believe it or not. So I don't even know, I forgot to look up how long people have been using cannabis-related products. At least hundreds of years, maybe even thousands. But the neurotransmitter, that, for instance, THC, which is tetrahydrocannabinol, which is one of the active ingredients in cannabis, the neurotransmitter that interacts with, anandamide, was only discovered in 1992. And I know some of you guys were born in 1992, but to me that literally sounds like yesterday. So this neurotransmitter was only discovered in 1992. And the name for it comes from the Sanskrit word Ananda, sorry if I mispronounced it, which means bliss. So what do these do in your brain? Well, they're both CNS and PNS, and they are involved in pain signaling, so the middle portion. So pain sensation, pain transmission or signaling, and pain perception. So they keep the pain from actually going to your central nervous system as readily. They're also, though, involved in working memory, which is like, oh, I just used cannabis and then now I go to the kitchen and I can't remember why I'm in there. Well, that happens to me without anything. But And then they're all also involved in eating and sleeping patterns, and then they're involved in pleasure and food rewarding. This tends to be really encouraging for people who are dealing with chronic diseases that make them not take care of themselves and eat. So for instance, cancer, those kinds of things, it's a decent treatment that both reduces pain and also makes it slightly more likely that you will take care of yourself. The receptors for these guys are called cannabinoid receptors. I realize those two don't look very alike, and they don't look like THC would actually activate the anandamide receptor, but I'll show you another picture. I'll show you where that they actually do that. Okay, so why are these so quick and effective to cause a response? So why, for instance, can somebody take a cannabis-related product, and if they don't take it orally, if they don't swallow it, eat it, ingest it, but if they use it by some other mechanism like vaporize it or something, why can it be a matter of minutes or seconds before it actually causes a response? Probably minutes. Well, part of it is that it is a lipid-soluble neurotransmitter. So once you get it across a mucous membrane into the body, it's moving into the central nervous system and into anywhere it wants to move in the peripheral nervous system. It's actually really, really quick. THC right here is an agonist for anandamide. It's a relatively weak agonist, but you have anandamide receptors all over the body. So here's THC and here's anandamide. They're really similar and they activate the receptor, and it activates it weakly and it falls back off, but there's just receptors. These cannabinoid receptors are called CB1 and CB2, and CB1 and CB2 are just everywhere, all over the brain. They're also present elsewhere in the body. So even if there's so many receptors, even a weak agonist can cause a really, really strong response. And so the biggest clinical connection is the whole THC. And then there are weak anandamide agonists also found in cocoa and chocolate. So like part of the reason you like cocoa and chocolate is because it kind of feels like cannabis to your brain. And this is just an interesting little aside. Anandamide, this guy right here, the actual neurotransmitter. If you take a petri dish of breast cancer cells that's actually growing, and you treat it with anandamide, it actually reduces the growth of the breast cancer cells. I'm not saying that it is a possible treatment at this point for breast cancer, but in the early stages, so you're still doing it like in vitro and not in vivo. Okay, so that's your endocannabinoids, and the only one that you're learning for the endocannabinoids is anandamide. Okay, so, and then the last little tidbit for today is just there are also some weird miscellaneous neurotransmitters that you would not expect to be neurotransmitters at all. I don't usually ask about them, but do we not both think it's weird that carbon monoxide will kill you if you breathe it? But carbon monoxide gas is actually released in the central nervous system as a neurotransmitter. We don't know what it does yet. And then nitric oxide, not nitrous that makes you giggle at the dentist office, but nitric oxide, that's a neurotransmitter that's being studied because this neurotransmitter right here is used in the peripheral nervous system, among other places, and it's used to cause erections. And so, not surprisingly, there's a lot of money being put into studying that one. Okay, we're going to stop there. I have just a little bit of stuff to finish in this set of notes, but I'm going to go kind of easy on the other stuff. You have a table. If you want to organize stuff on this table, the neurotransmitter table that I just showed you, you can use that, or you don't have to. If it's not in the notes, then you're not responsible for it. It's just people ask me about all of these drugs all the time, so I just went and looked them up and put them on a student version of this. So if it's not in the notes, but it's in the table, you can just ignore it if you want to. And then I don't want you to forget, because most of you haven't done it yet, I don't want you to forget that you have a quiz that is over synapses, neurotransmitters, and then the autonomic nervous system. I am, by the way, going to end up cutting out some stuff to make it easier for you guys to get through this, but you'll have to watch the videos to know which stuff I'm going to cut out. Okay, I will be talking at you in the near future. please participate in the discussion. Let me know what kinds of questions you have. I hope you're doing well. I will see you soon.